Hello, and thank you for joining and attending this webinar, Simple and Accurate Handheld Photosynthesis Analysis using the CI-340. My name is Dennis Fisher. I'm application scientist here at CID Bioscience, which you can actually see behind me in our beautiful facility in Camas, Washington, right across the Columbia River from Portland, Oregon. Now, I'm going to go to the agenda next. It is 8:01 though, so I'm just going to pause for a brief second to see if anybody else has trouble signing on, and give them a little time to do so. Now, if you are just signing on, I do want to say, if you look at the Zoom software, you have a couple different options. Now, one of them is a chat option, another one is a Q&A box. I will not be personally looking at the chat as I go through the presentation. Please, though, don't hesitate to use it. Our distribution manager, Susie Truitt, will actually be on the background posting links and answering a few questions that way. But the best way to get questions in our Q&A section is to actually put them in the Q&A box. That way, at the end of this webinar, I'll go through questions and answers, and I'll actually have them all queued up in front of me to answer them personally. Now, during this webinar, we'll first cover some of the general application areas of photosynthesis and respiration measurements, both in general and with the CI-340 photosynthesis system. Then we'll go over the system itself and assorted accessories to enable research. Then we'll show actually a video example so that you can see the instrument in the field. And then afterwards, we'll actually go over a couple of detailed examples of the CI-340 being used, including our special guest who will be discussing her project, Olyssa Starry. And again, just want to mention once more that the chat box, Susie Truitt, our distribution manager, will be handling that. I will not see it personally myself. If you have any questions, please put them in the Q&A box so that we can get to them at the Q&A section. I also put on, I had a lot of examples actually for this presentation. Only could cover a couple. So I actually put some appendix examples. We will be recording and sharing this presentation afterwards with all the attendees. And so you can get the presentation, the appendix examples afterwards, so you can look at through those as well. So one of the things I wanted to cover when it comes to photosynthesis study is that it's been pretty much evolving. Traditionally, plant scientists have squarely had control of photosynthesis measurements, including with such things such as phenotyping, carbon cycling dynamics, maturity and aging, characterizing stress responses, and relationships between photosynthesis and water balance. Now that has actually changed fairly recently. One of the uses we find with the CI-340 fairly frequently is actually as a teaching device. But we can also see CI-340 photosynthesis and respiration measurements as being used as part of the bigger picture when we're working with carbon credit assessments, solar technologies, and other scientific disciplines, including soil respiration and insect respiration. And due to these capabilities, its portability, the CI-340 has been utilized extensively in many of those aforementioned areas. To show its use both as a research and educational tool, I've put links actually in this slide, and Susie will also put them up in the chat. So first link, the research tool, actually just goes to our website, cid-inc.com, where the post papers using the research. In this example, I've used effects of starter nitrogen fertilizer on soybean roots, differential early stage water relations for when it comes to tomato and salt stress, and the effects of ozone treatment and light color on photosynthesis and yields of lettuce, just to show not just a variety of different applications, but also from China, India to Poland, just how the CI-340 has been used across the world. Now the second link is often used, is showing the educational purposes. Now it's thanks to a hard work of our one of our partners, Simone Whitecloud, She's created the teacher training measuring photosynthesis and respiration real time with an ERGA using the CI-340. This is used as a method for teaching photosynthesis and respiration. Actually, anywhere they use this particular link. That will also be in the chat, thanks to Susie. So, to get straight to what the instrument is, the CI-340 handheld photosynthesis system, providing essential utility in the lab and field. It's light, it's handheld, and doesn't require a backpack of gear. The instrument is really just field ready, as you can see there, with less than 3 pounds. Now, the CI-340 does take non-destructive, open or closed system measurements, and it utilizes a variety of sensors, including infrared or ERGA carbon dioxide analyzer, a humidity analyzer, PAR sensory data, multiple temperature data, including sensors to measure the leaf temperature, air temperature, and the internal temperature. Now, all of this data is able to give you information such as photosynthetic rate, determining by measuring carbon dioxide before and after it enters the leaf chamber, and to calculate the rate of carbon dioxide assimilation by a known leaf area. Transpiration rate, determined by measuring water vapor before and after it enters the leaf chamber to calculate the rate of water vapor flux per one-sided leaf area. Stomatal conductance, calculated by measuring transpiration rate as a function of leaf temperature. And all of this is done automatically by the instrument, and it spits out the data both on the instrument itself as well as to a computer you connect to. Now, the CI-340 is easily operable as either an open or a closed system. In open system measurements, the incoming gas is drawn from ambient or control module altered air, and the exhaust returns to external atmosphere. During closed system measurements, the air is recirculated from the exhaust back into the chamber. So the CI-340 is designed to measure both absolute, also and differential readings. In absolute mode, the CI-340 measures gas concentrations from a single source. And for differential measurements, the concentration of gases from both the inlet of the chamber and the chamber exhaust are measured. Now, due to the large variety of research, we have actually over 11 chambers for whatever application you might require and may be studying, as well as 4 additional modules that are optional to give you this optional flexibility in your studies. Now, this wide selection of leaf chambers does fit a large variety of leaf types, sizes, stages of maturity, and actually quite more than leaves. They work, again, either as open or closed system chambers, and some also accommodate a stem entering the chamber while sealing around it. Open systems are configured so that the air is continuously passed through the leaf chamber to maintain carbon dioxide in at a stable concentration. And measurements of photosynthesis and transpiration are based on the differences between carbon dioxide and the relative humidity in the air. The rate of carbon dioxide uptake is used to assess the rate of photosynthetic carbon assimilation, while the rate of water loss is used to assess the rate of transpiration. And this is determined again based on the leaf area. Now in the closed systems, a leaf or other matter is enclosed in the chamber itself. It's sealed to avoid gas exchange with the atmosphere, and the rate at which the carbon dioxide and relative humidity concentrations changes in the chamber are monitored. This is determined based on leaf area and the leaf chamber volume. And complete equations actually for photosynthesis and transpiration are all available online at our website, again, cid-inc.com. Now let's go over a few of the chamber options that we have available to us. The first one, so we have our standard leaf chambers for measuring trees, shrubs, grasses, either small broad leaves, small regular leaves, or even grasses and grass-like leaves. We call these LC1, LC2, and LC3. Now, for open system measurements of succulents, conifers, either small needled or large needled, we have LC4 and 5. And actually those flaps that you can see there, the little kind of orangish red tomato color, are actually allowing the needles to be placed non-destructively in the chamber so that you can measure it even while having only part of the plant there. Now the LC7, 8, 9, and 10 are actually for measuring leaf litter, fully contained materials, and much more, including in one particular case, sea cucumbers. In the inside, you can kind of see here the internal fans that run when you attach the CI-340, and that's connected to allowing for measurements to be taken in more of a homogenized atmosphere, regardless of the increase in the volume. And the major difference between these is, as you can see in the top right corner, that is the LC7. It's only a quarter liter, and these go up to a full liter chamber. Now, for more miscellaneous and oddball type of chambers, we created the LC11, which is for cactus leaf chambers. You can see in the top right corner there. We actually created a soil respiration chamber as well, which is this large one right in the middle. And we actually also have, just in case you want to create your own type of chamber, we have the CI-301cc canopy chamber attachment. And it's basically an interface between any chamber you create and the instruments, allowing you to design and adjust as you will. Now, we also have 4 optional control modules for use in the field or lab for assessment of plant response and variable conditions. The light module, the CI-301LA, allows researchers to adjust light intensity received by the leaf in the chamber. The second one, the 301AD, carbon dioxide and water control, enables researchers to set or adjust carbon dioxide and water concentrations in the chamber in order to investigate related physiological changes. The third, 510CS, temperature control, enables researchers to adjust temperature in the chamber to evaluate fluctuations in the rate of photosynthesis relative to high or low temperatures. And then finally, we have the chlorophyll fluorescence, which measures fluorescence simultaneously with photosynthesis to provide researchers with the information about changes between photosynthesis efficiency and heat dissipation from the leaf. Now, again, these are all optional modules. You could use 1, 2, or all 4 at the same time. And they are frequently not even necessary for most of the studies that are utilized, but they are very useful if you would like to see how your study reacts under different conditions. So to more effectively show the CI-340 in action, I'm going to turn now to Susie to show off a brief video we've created demonstrating the instrument.
The CI-340 Photosynthesis System is a portable, field-ready tool that can measure photosynthesis, respiration, transpiration, stomatal conductance, photosynthetically active radiation, and internal carbon dioxide. 10 different customized chambers are available that can accommodate any leaf size, including broadleaves, grasses, conifer needles, and cacti, with an optional chamber to measure soil respiration. Lightweight and durable, the CI-340 was designed to be easily utilized in the field. To set up the instrument, first attach the leaf chamber. Then attach the temperature sensor. Next, attach the photosynthetically active radiation, or PAR sensor. After installing the rechargeable battery, you can then power on the instrument, attach the leaf chamber to your leaf specimen, and after a brief warm-up, the instrument can begin collecting data. The optional accessory modules allow researchers to control carbon dioxide, water vapor, temperature, and light intensity inside the chamber, as well as measure chlorophyll fluorescence. The CI-340 is based around gas exchange happening at the leaf surface, including both carbon dioxide uptake and the release of water vapor. The concentration of gas passing into the chamber and leaving the chamber can be used to determine net photosynthesis of the leaf. Once your measurements are complete, information is easily migrated from the instrument to the computer via a CSV file, making the jump from data collection to data analysis seamless and stress-free. The CI-340 photosynthesis system gives researchers the power to collect real-time data with the push of a button. Its lightweight design and customizable accessories help scientists worldwide conduct groundbreaking research.
All right, fantastic. And now that we've kind of covered the video with more of field examples, actually let's turn now to a couple of real-life examples for more in-depth of how the CI-340 can work. First, we'll start out actually with Benoit's group, the Eastern Townships Forest Research Trust, and their use is the 340 in poplar research. They have used the handheld photosynthesis analyzer as well as the soil respiration chamber accessory to study both soil respiration rate in mature hybrid poplar plantations in southern Quebec and Canada. So here first, the materials and method of this particular project. Soil respiration was measured at 3 different 14-year-old hybrid poplar plantation sites located in southern Quebec, Canada. This is Brompton, La Patrie, and Melbourne. And the experimental design contained 27 plots, so that's 3 sites, 3 poplar genotypes, and 3 blocks per site. So in each plot, soil respiration, including root respiration, was measured in situ using a CI-340 ultralight portable photosynthesis system. And they also did 3 sampling times, so they're selecting late May, late July, and late October. So respiration was measured by placing the CI-340 chamber on the soil surface for about 2 minutes time period during which the carbon dioxide production was measured. And this procedure was repeated at 5 random sampling points in order to account for soil heterogeneity within the plots. Now, soil temperature measured within that chamber, you can see there, and air temperatures were recorded simultaneously during respiration measurements. Soil respiration rate and soil temperature, air temperature were averaged for each sampling time in each plot. Now, the next slide, I just want to show some pictures of the work in progress. So the photo on the right, this is actually a photo of a mature hybrid poplar plantation near the region of La Patrie, Quebec. And you can see that there's a very large amount of leaves on the ground. And this is actually the highest altitude experimental hybrid poplar plantation of this entire climate zone. And then to the left, you can see some of the root testing, leaf litter testing, going down far to access actually both of those materials. Now the results, the respiration rate followed the same trend at the Brompton and La Patrie sites, with lower respiration rates observed in the spring and the fall, with a peak during summer. However, respiration rates were much higher at the Melbourne site in late May compared at the other 2 sites, and not different from the respiration rate measured in July. At Melbourne, air and soil temperatures were also higher during respiration rate measurements in late May. So, and then the images at the bottom, you can see that the handheld photosynthesis system was using a tripod mount just to make for convenient measurements. Now, they actually enjoyed using this instrument so much, it's being continuously used. So the Eastern Townships Forest Research Trust is using the CI-340 for things like a study on the utilization for multi-species plantation in the agricultural landscape. And these species include like hybrid poplars, red oaks, bur oak, red ash, white pine. And as well as in comparing kind of different silvicultural treatments arranged in split plots, including things like black plastic mulch and control with like weed plots. The second example is I'm going to use go all the way to a little bit further south, Sedum Genera and the University of the West Indies. Now, to give a little bit of a background to this particular study, they wanted to research the regeneration ecology of a moist forest over limestone in Cockpit Country, Jamaica. Now, Cockpit Country is pockmarked with steep-sided hollows as much as 390 feet or 120 meters deep in places which are separated, as you can see in those pictures, by conical hills and ridges. And the objectives of the research were to collect and assess data on forest dynamics, assess major trends and relationships on those dynamics and different environmental variables, and assign tree species to guilds based on the plant ecophysiology and dynamics data. So there were a few challenges to the project, as I connotated. The limestone karst topography had very loose surface rocks. They have an average slope incline of actually over 45 degrees, requiring backpacking hikes up to 4 hours just to reach the locations for testing. And there's also like daily convectional rainfall where the land warms up, heats the air above it, causes the air to expand and rise. And as it cools and condenses, you're getting pretty much a constant afternoon rain. And of course, they needed an instrument that had a suitable power source for their equipment. Here in the picture, you can see actually to the right, you see a CI-340 fixed to a tripod and right on a very steep incline. Now, there are several advantages to using the CI-340 for this project, including extremely portable, light, and you can actually do on-site calibrations in the field. Now, on the left here, we see the CI-340 performing warming up procedures prior to measurements. And on the right, it's actually the light module attached doing the measurements required. And then finally, here we see the CI-340 with a light module attached again, and a modified setup so you can have constant carbon dioxide going into the instrument just to reflect a varied forest floor carbon dioxide changes throughout the day. Now next, we will hear from our guest speaker, Olyssa Starry, discuss her testing site and fascinating research into the green roof dynamics.
Hi, everyone. I hope you can hear me. I guess I'll get a message if you can't. So I just want to introduce myself. I'm Olyssa Starry, and I did my dissertation at the University of Maryland in College Park, and my degree was in plant science and landscape architecture. I'm currently faculty at Portland State University, where I teach in the Honors College, and as you heard, I conduct research using green roofs as my system of interest. So I test urban ecological theory using green roofs as model systems. And there are a couple of ways that I use the CI-340 in my research. I'm going to just share a picture with you of a green roof that we, oh, maybe I don't want to stop other screen sharing, so I'm not going to share a picture of a green roof that
no, you go right ahead.
I can go right ahead. Okay, I'm going to share a picture of a green roof that I use, or that I have worked on in Portland. This is the East Multnomah County Courthouse building. So anyone can visit this roof, and it has a great view of Portland. I highly recommend it. Anyway, there are two main areas that I use the CI-340, or that I would use the CI-340 for. One is to study the green roof water balance. And so, especially here in Portland, when we get all that rain in the winter, really the only way to dry the soil down and achieve stormwater management with these systems is through the process of evapotranspiration. And so this CI-340 can not only help with stomatal conductance measurements, but it can also measure rates of photosynthesis of the plants on these green roofs. And as you can see, there's quite a variety of plants used. Here, there's a lot of grasses, which are C4 plants primarily. But another genera that is typically installed on green roofs is the Sedum genera. And as many of you are aware, Sedum are primarily cam plants, which means that they open their stomates at night so they can conserve water during the day. But what a lot of people don't know is that Sedum are facultative cam species. So that means that they can switch in and out of this water conserving mode of photosynthesis. And a key hypothesis of my work is that they're actually not in CAM when water is readily available. And so that even these succulent species can contribute to the green roof water balance and stormwater management. And so we can collect the data we need to test this hypothesis using the CI-340. And another area of my green roof research is addressing the question of how these systems contribute to air quality management. And so, as you know, a lot of the HVAC intake elements are located on the roof. And so what some of my colleagues in the engineering department at Portland State, Elliot Gall, for example, and I are interested in knowing is how does your roof choice affect the quality of the air that's coming into your building? And, you know, given that people spend, I think it's upwards of 90% of their time indoors, it's really important to understand the influences on indoor air quality. And so we have a graduate student who was able to determine that there is a ratio of CO2 and water use by plants relative to the rate of ozone deposition. So you can predict ozone deposition onto a green roof surface if you know something about stomatoconductance and CO2 uptake. So we're going to use the CI-340 to validate and verify some of the measurements that we've been taking on ozone deposition and some of the ozone deposition modeling that we've been using. And I would just finally just like to add that some of the reasons why the CI-340 really works well for our roof or for our research is because it is very portable. It's very easy to use. There is wonderful customer service. And it's really also, and finally, one last comment I'd like to make is that it's really valuable to me as an educator. So this summer, I led a workshop for some junior transfer students in the STEM fields, and it was sort of a last minute thing. I called up the people at the customer service and they helped me get our instrument calibrated in time. They directed me to all of the manuals that are helpful for understanding how to download the data, all of the technical information. And I'm here to tell you also that we were able to walk the students through the steps in the manual for collecting data in a research setting, and they were able to do it without any obstacles. And it was really valuable also to be able to collect real research data with these students instead of giving them, you know, a canned problem set. So I would be happy to answer any questions about this work or my experience with the instrument, but unfortunately I have to go teach class. So my email is just my name, my first initial and my last name, Starry @pdx.edu, and it's really easy to look up online as well. So I would be happy to answer any questions via email, or you know, people can send me questions through this service. Thank you.
Thank you very much, Dr. Starry, for that presentation. I'm going to go ahead and take back the screen now. There we go. And now that we're towards the end of our presentation, we're actually going to go into questions and answers. But because, you know, we're really glad that you were able to attend, we have a form at the top here. You can see the tiny URL we have for if you wanted a quote for the CI-340. We are offering actually for all attendees the first year of maintenance and warranty absolutely free. So that's $1,280 value. And again, please put all questions into the Q&A box and we'll get to them accordingly. Just keep your eyes on the chat as our distribution manager, Susie Truitt, will be posting links so you can see the slide for easy access for the top where it is the quote form. On the left, there's a link actually to our website so you can find more information about the CI-340 and the other instruments that CID provides, as well as if you wanted a consultation individually with myself or our other application engineer who will be on later on today. Katrina Tsao, and you can use that to have a one-on-one session with us to discuss your personal application. Now I am going to go to this Q&A box. All right. All right, so the first question we have is, does Arabidopsis and such small plants measurable with the instrument? And definitely. In that case, you can actually use either open or close with some of these smaller chambers, I would suggest. Or if you wanted to get the entire plant into one of the larger chambers, you'll be able to use that as well. The next question is, as a large number of measurements, what about the calibration data of the different sensors? And all of that actually is going to be available and print-offable from the instrument itself. So you can attach to a computer, get the information as you kind of saw in the video. It usually comes in the CSV format. And there we do give step-by-step instructions for how to do the calibrations. And you can also see those on the instrument itself. So you can do it in the field. Now, how long is the process for measurements? That actually is a really good question. It really depends on the environment that you're looking at. Normally, if you're doing like a continuous flow across, it can take a couple minutes. But if you're trying to do, usually if you're doing a setup where you have, say, like a large container, or you're doing respiration over, say, 5 to 10 minutes, it could take as long as that. But you do start getting data right away from measurements. So it really just depends on your particular application, how long it's going to take. The next question is, can we use the LC7, 8, 9, or 10 to make whole plant measurements? Those, if you recall, are the ones that are just a large chamber with the fans inside, and that is definitely the case for those. Now, normally we created them originally for leaf litter, but quite often cases that they're used for things like whole plants, insects, and I think, as I mentioned, the coolest one I ran into was a sea cucumber. The next one is, can this be used to measure canola silique photosynthesis? And I mean, honestly, I would have to say, I would guess yes, but I would want to know a little bit more about the application or how you're trying to measure canola before I could give a definitive answer to that. But because you are measuring carbon dioxide in, out, and transpiration rate, the water rate, you should be able to get the data you need. The next question is, it's talking about chlorophyll fluorescence. So as mentioned, we do have a module for chlorophyll fluorescence and measuring that in comparison to the photosynthesis rates and heat dissipation. So you can kind of get all three of those different uses for chlorophyll and the energy that's being provided, and find out, you know, where that energy is being shunted. The next question is, can you check the relative humidity in the chamber? And that's yes. In fact, we have a sensor for that, just to make sure that the humidity chamber, humidity outside are you know, either equivalent, or if you know they're not, where they're at. Next question, is it possible to score hydric stress? Yes. Yeah, you're actually, if you're measuring, you're both measuring water rate, flow rate, you're able to get changes in those and get some information on stress of the plant. The next question is, can photosynthesis be measured while the plant is growing, specifically microgreens, which are vegetable seedlings? Yes, you can measure. It does measure low enough that you can actually pick up transpiration and photosynthesis rates in very small amounts. We definitely would suggest using a smaller chamber, of course, just so that you keep the atmosphere as microclimate as possible. The next question is, how can you use fluorescence and light modules simultaneously? How do you mount the modules in the chamber? It's a little bit different depending on the chamber you're using. If you're using the module for like the smaller chambers, you actually would mount them just on either side of each other. But if you are using one of the larger chambers, that would be a bit more difficult, and it's a little bit more intensive process to set that up. The next question is, can you also generate light response curves for different varieties? And yes, in fact, so the data that's given off is in a CSV file, so it's actually very easily to in either Excel or a program, if you're choosing, to create graphs out of the different curves and the different varieties and the information from there. How fast is the instrument measuring photosynthesis? I guess the way to best put that is you have a flow rate of 100 to 1,000 centimeters squared per minute. It really depends on the flow rate, again, your application, but it starts measuring as soon as you turn it on and attach it to the space, to whatever plant you're looking at. The next one is, why not include soil sensor temperature directly on your device to use with the soil chamber soil respiration? And then the soil chamber soil respiration, you are getting the chamber temperature of the soil, and that's why we created the chamber that way. So when you stick it all the way into the soil, the sensor at the end is actually measuring what that soil temperature is. Now, of course, as soon as you take it off, I won't actually be working in that regard, but yeah, it does provide that information. Is there any possibility to control the carbon dioxide concentration inside the chamber? Yes, that's actually what the carbon dioxide and water control module would be for. You'd use that attachment and dictate the carbon dioxide concentrations according to that. Actually, although you can do some jerry-rigging as well, the way the instrument's designed, as you saw in the Jamaica example, they actually just put a line into a carbon dioxide cartridge, and ran with that. Can you use the CI-340 without a module? Yes. Actually, the modules are all optional. There is no need for any of them unless your specific application requires that specific control and or photosynthesis fluorescence measurement. Besides that, it's measuring for photosynthesis, net photosynthesis rate, transpiration, respiration. How long can the battery last in the field? Very good question. We actually usually suggest if you're using the battery intensively, you're going to have about 6 to 8 hours. And we do provide actually additional batteries, car chargers, and a wall mount, so you can charge it pretty much anywhere you want to go. Let's see, the next question is about batteries again. The next one is, how can I calibrate the temperature sensor in the CI-340? That actually is an extensive, and it's in our manual how to do so, and I'll be happy to do that in either a side presentation, but that would take a couple minutes to go through it step by step. What kind of various info light intensity module provides? So the light intensity module, actually, it's you're going to get, let's see, red peak wavelengths. So it's 660 nanometers to, and then at blue peak wavelengths, it is 470 nanometers. So those are the LEDs on the module. It gives you an output range of 0 to 3,000 moles, and micromoles, and then the precision is a par like plus or minus 15 micromoles meters per second. So what you're able to do is take that data, control the light measurements, and then just see how your plant, insects, depending on the project, will react either through photosynthesis or respiration rates during those different changes of light. Usually this is a kind of getting a more rapid idea of like dust conditions, different wavelengths, and how if a plant is going to increase respiration, decrease respiration, increase photosynthesis, or decrease photosynthesis. What is the time frequency for the calibration? Which is actually a really good question. We actually suggest that with the ERGA sensor, the ERGA carbon dioxide sensors, we want that, we would suggest every 3 months, however, 6 months would be fine. And for the oxygen sensors, pretty much the same as well. The next question is, how long does it take to calibrate the unit? It takes about half an hour to do so, usually a lot less, but I'm thinking in the field, if you're trying to work with things that are a little bit unwieldy, about half an hour. If you're doing a less intensive and have actually a lab setting, it would take like 15, 20 minutes. How long can the instrument continuously acquire data, and what are the smallest time intervals it can record? I'm just wondering if you could do a 72-hour continuous testing. And yes, actually, so we do have another like a wall mount. Instead of running on battery, you just plug it into the instrument, and you can run it in more of a lab type setting and just have it keep going on whatever chamber or that you may design or that we provide and get all the data. And so each one of those time intervals, you can do it continuous. You can actually do it for just like 1 minute and the next minute, though I would actually highly suggest continuous. The data is very useful. How long does it take to construct ACI curves? That's a good question. I am not sure. That would be taking the data that would off the instrument. So it really depends on what programs you're using to create those curves. You do have the data available to do so, though. The next question, to measure photosynthesis in pecans, which are the values to set up device before you start taking lectures? So actually, to measure photosynthesis in pecans, you can just set up the device, calibrate, have it run, get used to the environmental air, and then you're ready to go. So if you have leaf material, if you have small like pecans, you can use probably the small chambers that we showed, and you just put them in, close it down, and start running, and you'll start seeing that data show up quickly. Can we regulate the relative humidity? Yes. Again, with the carbon dioxide and water chamber, you'll be regulating relative humidity in the chamber, but that will be using with that optional module. The next question is, if you bring competitor, are there any difference in functioning between the CI-340 and the Lycor 6400? The biggest actually differences between them, I would say, are the CI-340 is much lighter, and all of the features, there are a lot of features in the Lycor 6400, I'm not going to lie, but you don't have to use them all, and it does require lugging all of that around with you in the field. The CI-340, a lot of the features you can buy optionally in those optional modules, and that way you're not regulated to that. The other thing is like the information is a lot easier to access, so when you pull the data off into a CSV file, you're able to take that data and do whatever you want with it without any secondary cleansing. Next question is how much data can be stored on the CI-340. It actually uses an SD card to store a lot of the data, as well as a serial bus. So really you could store, and I think the earlier question was 72 hours worth, very easily. I would say at least a couple weeks worth of just continuous data if you would like. Normally I've taken it off well before that. And is it possible to update the instrument software? Would that require to be sent to CID Inc? The instrument as it is right now, yes, you can actually update the software via any kind of updates we push to our website, which we actually have on cid-inc.com, a software section. And for any of those potential dates, we'll actually send out whoever has purchased one a notice to say that this update is coming around, and give you the option of whether you want to apply it or not. All right, we are running well over time. So what I'm going to say is, a lot of fantastic questions, and I'm going to close it out now. But if you have any questions in the future, please do not hesitate to send them to myself. Again, on the screen, you can see that you can schedule a consultation, or just check out our website. There's a lot of material there to go through. And again, thank you very much for joining. And if you would like to get a quote, please just use the link at the top to get a quote.
Thank you again for joining us. In the chat box are the links if you'd like to request a quotation, or if you'd like to speak to one of our application scientists with your specific questions about your application. Thank you again for joining us.